So this lecture we will be discussing about the antigen. Now, what is antigen? Antigen is any substance which induces a specific immune response, which is leading formation of specific antibody or reactive T cells. That means, which is what is the most important thing, that it should challenge the immune system. It is also called as the immunogens. Why it is called as the immunogens? It is called as immunogens because it is challenging the immune systems and it is making the immune system engage in doing its job. So most important characteristic is immunogenicity and immunoreactivity. See, immunogenicity means ability to produce immune response, and immunoreactivity means ability to combine with its antibody. So not necessary that all antigen will have the immunoreactivity. Some antigen can just bring about the immune response and they can be killed by the T cells. So we had all discussed all these things into the T cells and cell mediated immune response. So antigen have certain property, means we cannot say any foreign substance is an antigen. It should come into the all these four criteria. That time only we can say that it is an antigen. So first thing is the foreignness. It is most important thing that antigen must be foreign to the host. Foreign to the host means it we are taking it or it is trying to enter in our body from the outside. Any inside substance means our host system cannot be the antigen. If your immune response is considering self part or self cells as the non-self, that particular time it will be called as the autoimmunicity. So antigen must be foreign to the host. Antigen from related species are less antigenic than of unrelated species. What it means that related species are more antigenic. Antigenic, I told you, having the property to initiate the immune response. So the substances which are from the same species. Example, this we have taken the blood from the human has taken blood from the another human. It will not produce that much antigenic response than the other species. Understood? So this is something about the related species. Human and human are the related species. So proteins also, if we will talk about, plant protein will have the good antigen into the animal. All foreign substances cannot elicit immune response. Example, carbon particle can invoke the phagocytosis but will not stimulate the antibody production. Antigen from one organism will invoke greater immunogenicity in that are phylogenetically distant from each other. Example, the human serum albumin is non-immunogenic to the human. Just now I have given you example that if the blood is taken same way, if the albumin is taken from the other human. See, for taking the blood, we have the RBC, it's antigen. But when we take the albumin, irrespective of the blood group, we can pass on. Plasma therapy works on it only. So, but that serum albumin, if injected into the rat, it will give the very harmful effect to the rat. But if same thing will be entered into the monkey, it will show the less immunogenic because phylogenetically monkeys are related to us as compared to the rats. Phylogenetic matlab means evolutionary. When we talk about the evolution, so we are very close to the monkeys as compared to the rat. So if our serum is transferred into the serum albumin is transferred into the monkey will not show so much antigenic response. But same thing will, if injected into the rat will show more and more immune response. May other species other than mammal, if you will put more and more immune response, so it is showing that it is having the more and more foreignness. but not all foreign particles. Whatever food you are eating, it is foreign to your body, but it is not at all eliciting any immune response, although it breaches the barriers, but it do not produce. Why it is allowed into the body? Any food which is harmful for you, suppose you are eating something very stale, or it is having some pungent smell, your body is not allowing to eat that thing. Suppose if some food is there and it is having the foul smell, your nose, we have the sensory system, your nose will tell that this food is spoiled, it is not good for you. Then suppose if the food is not having the pungent smell, but still it is having something which is bitter taste or something, so your tongue will give you stimulus, don't eat, it is not good for you. So even if the food is spoiled, we have the challenges. Means our body is well organized to take care of all these things. So and even if you have tasted it, you know it is not good, but X, Y, Z reason you are very hungry, you don't have anything rather than this to eat. If you will eat that time also, that whatever foul substances you have taken will be taken care by the immune system, and you will get the vomiting. Understood. So this is about the foreignness. Then next category is the molecular size. A very small particle will not initiate any immune response. Right now, very small means we have the unit delta. So any substances which is having molecular weight less than 10,000 molecular weight less than 10,000 Dalton will not be able to produce the effective immune response, but it does not mean that it will not be able to produce, because the small molecules such as insulin, which is having approximate 5,000 Dalton weight, and glucagon 4,600 Dalton weight, can function as the immunogen, but immune response will come very minimal rate. So anything which weighs more than 10,000 Dalton and having means molecular weight approximate between the range 10,000 to 1 lakh will give the good immune response, but the best results can be seen on the larger molecular weight. Then heterogeneity and the chemical composition. Heterogeneity means it should have the protein and polysaccharide have the more and more good quality of the immune response, and you will find that the lipid and nucleic acid, they do not produce the good immune response. Artificial homopolymers are non-immunogenic regardless of their molecular size, whatever big they are. So when we talk about the protein, so proteins are better antigenic when compared to the polysaccharide. Means see, we have the biomolecules as protein, polysaccharide, lipid, and nucleic acid, right? So out of this, the best immune response can be seen when the antigen is protein in nature. Okay, and if the protein and polysaccharide, both are, polysaccharide can also elicit the immune response, but when if same molecular size protein and polysaccharide is there, you will find that protein is giving the good type of immune response as compared to the polysaccharide. Then insoluble will also give good immune response. Aromatic amino acids, amino acids like tyrosine, phenylalanine, tryptophan, they enhance the good quality of the antigenic response. In addition of the tyrosine and phenylalanine, if polylysine homopolymer or addition of galactosamine will add it, we can see the profound effect on the immunogenicity. So heterogeneity is required. If the antigen have the heterogeneity and their chemical composition are comprised of the aromatic amino acid, they will show more and more immune response. Next thing, it should have the degradability. Degradability means its the ability of antigen to process and present with the MHC molecule. We had already discussed antigen processing and presentation. So for the processing, it should degrade until unless it is not chopped by the enzyme, so it cannot be presented on the MHC molecule. So development of both humoral and cell-mediated immune response requires the interaction of immune cells with antigen that has been processed and presented together with the MHC molecule. So for that particular region, large insoluble macromolecule will give more immunogenic response as compared to the small soluble one, because larger molecule can be readily phagocytosed and processed. Macromolecules are means that cannot be degraded and cannot be presented on MHC molecules will not show the immunogenicity. So substances which are rapidly broken down by the tissue enzymes will also not show any antigenic, because tissue and it should have the degradability, like it should be broken by the antigen presenting cell phagocytosis. Example, stainless steel pins are there, stents are there, pacemaker, plastic joint. These all are inert, immunologically inert, so they cannot bring up any type of the enzyme degradation. That's why they cannot be killed. Now, next thing we will learn is the antigenic determinants or the epitopes. Immune cells cannot interact or recognize the immunogen molecule, which is having the smaller means this molecular weight less than 10,000. And the next thing, that all part of the antigen's body is not the antigenic. It have the special sites or special macromolecule which is called as the epitopes or the antigenic determinants. Means a whole bacteria is there. So whole bacterial body is not immunogenic, will not elicit any type of the immune response, but it will have some sites which we have learned in antigen presentation and processing, that the antigen presenting cell, what they are doing, they are chopping the antigen and they are flagging some parts. So these particular parts are the epitopes, are the antigenic determinants. Means they are recognizing, yes, these are the sites which is eliciting the immune response. So the site which eliciates the immune response, or which is bringing up the challenge to the immune system, is called as the antigenic determinants or epitopes. So these epitopes are immunologically active region of immunogen which binds to the antigen specific membrane on the lymphocytes, or it secretes the antibody. So whatever antibody is being formed into the body, it is formed against that particular part only, which is immunogenic, not for the whole bacteria. B cells and T cells can recognize different type of epitopes on the same antigen. Means one antigen is there and it is having the many region which is antigenic. So B cell as well as the T cell, they can recognize that one antigen may have more than one epitope. It is not necessary a one bacteria is there, so it will have only one epitope. It may have 10 epitopes. So this B cell and T cell can properly differentiate between all these epitopes. Haptens or incomplete antigen, as we have learned that any substance to be called as antigen have the molecular weight minimum 10,000 Dalton. So if it is very small in molecular weight, that cannot, although it is harmful for the body, but due to its very small molecular weight, it will not be able to bind with the carrier. It will not be able to bind with the antigen presenting cells. So for that time, these immunogens are bind with the carriers such as latex or polystyrene particles. Now, you might be thinking that how body will know to bind it with the carrier. This is for the artificial purposes. Suppose one antigen is there. Okay, so one bacteria is there and we want to design the antibody against that particular bacteria in the human body. So what they do, they just cut off the antigenic region of that particular bacteria and they bind it with the carrier molecule. So what will happen? This carrier molecule is inert for the immune system. So antigen against antibody will be produced in our body, and it will remain forever. It will form the memory cell. So this way the vaccines have been formed. Now, this antibody elicited by the hapten carrier conjugate can specifically react with the haptens even in absence of the carrier particle. So, if once the antibody is designed, so this hapten, even if it will come alone or it will come with the whole form, so it cannot be left by the immune system. Once the antibody is been designed, they will catch hold and they will kill even if the carrier particle is absent. So this is the general idea. When soluble molecules are used as a carrier, this molecule can also be immunogenic to the host. So most of the time soluble molecules cannot be used. They are generally using the substances which are inert. So this is all about the antigen, and next lecture we will be talking about the antibodies.